Dear students, let's continue Chapter 2, Windows of Knowledge. Today we shall discuss about the regulation of light in the eye, then formation of image in the eye, and also we shall discuss about retina and photoreceptors. First, let us learn about the regulation of light in the eye. This is a very simple topic, so let us move on to the topic. So here you can see this is an eye. Then the pupil of eye is represented here as enlarged diagrams. Then this is the condition of pupil in dim light and this is the condition of pupil in bright light. So here you can see the size of the pupil is larger. This is the pupil and these are muscles. So here the size of the pupil is larger in dim light. Then here you can see in bright light the size of the pupil is smaller. So the size of the pupil is regulated by two types of muscles. So here you can see there are two types of muscles. This is radial muscles and these are circular muscles. Here also you can see these are radial muscles and here this is circular muscles. Then in dim light, what is happening to the muscles? In dim light, you can see the radial muscles. So these are the radial muscles. In dim light, these radial muscles contract. In dim light, the radial muscles contract so that the size of the pupil increases. Then in bright light, you can see the circular muscles contracts. So as a result, the size of the pupil decreases. So in bright light, the circular muscles contract and as a result, the size of the pupil decreases. So in dim light, the size of the pupil is larger and in bright light, the size of the pupil is smaller. Now let us learn about the formation of image in our eyes. Now look here, this is the structure of eye. And then this is the lens that present in our eye. This is a convex lens. And then this is the object and here you can see the image formed. Now you already studied in your physics class about the image formation in convex and concave lens. So the same process is taking place in our eye also. So here you can see this is the object. Here the object is a tree. And then the light rays that falls upon the tree is reflected back and it enters through our eye, through the lens and then it focuses an image on the retina. So you have already studied about retina while learning about the parts of eyes. So the retina is located here. Retina is a screen on which the image is formed. So here you can see this is the image formed on the retina. Now look at the image and the object. Can you see any difference between object and image? Yes, you can see the size of the image is smaller while comparing with the size of the object. And then what is the other difference? Here you can see the image formed is in inverted position. So here the object is in upright position but the image formed is in inverted position. And also we can see a real image is formed here. Children, are you remembering about your practical experiment of image formation using convex and concave lens in your physics lab? So while doing that experiment, for getting a clear image, you can adjust the lens. You can move it forward or backward for getting a clear image. You can adjust it according to the distance of object. So our eyes is capable of viewing distant objects and also nearby objects clearly. So who is doing all these adjustments inside our eyes? There are certain muscles present in our eyes. That is ciliary muscles and ligaments. So the ciliary muscles and ligaments helps for adjusting the lens or the focal length of the lens according to the distance of object. So now let us see how the image or how the lens is adjusted for viewing a distant object. So here you can see a distant object, that is the tree. For viewing a distant object, you can see the ciliary muscles in our eyes relaxes and the ligaments stretches. So where is the ciliary muscles and ligaments? Here you can see the ligaments and here it is the ciliary muscles. So the ciliary muscles relaxes and the ligaments stretches. So as a result, the convex lens become thinner. The convex lens become thinner. So when the convex lens become thinner, we know that the curvature of the lens decreases and as a result, the focal length of the lens increases. So when the ciliary muscles relaxes and the ligaments stretches, the convex lens become thinner and as a result, the curvature of the lens decreases and the focal length increases. So the focal length need to be increased for getting a clear image of the distant object. So here you can see when the focal length increases, we will get a clear image on the retina of the distant object. And now let us learn how the lens is adjusted for viewing a nearby object. So for viewing a nearby object, the ciliary muscles contract and the ligaments relaxes. So when the ciliary muscles contract and the ligaments relaxes, you can see the convex lens become thicker. So here the convex lens become more thicker. So as a result, the curvature of the lens increases and the focal length decreases. When the ciliary muscles contracts and the ligaments relaxes, you can see the curvature of the lens increases and as a result, the focal length decreases. So the focal length need to be decreased for getting a clear image on the retina of the nearby object.
Now we shall learn about retina and photoreceptors. We already know that the receptors present in our eye are called photoreceptors. These photoreceptors are present in the retina. Photoreceptors consist of rod cells and cone cells. The name rod cell and cone cell can be related to its structure. You can see a rod shape on rod cell structure and a cone shape on cone cell structure. The visual pigment present in rod cell is known as rhodopsin. This pigment is formed from a protein named opsin and retinal. Rod cells helps us to see the objects in dim light and these cells cannot detect color. The pigment present in cone cell is known as photopsin. Photopsin is also known as iodopsin. This pigment is also formed of opsin and retinal proteins. There are 3 types of cone cells in our eyes that helps to detect primary colors including red, green and blue. Cone cells helps in color vision.
Finally, let us learn about the chemistry of vision. So here you can see rhodopsin. Rhodopsin is the pigment present in rod cells and photopsin is the pigment present in cone cells. So in the presence of light, both this rhodopsin and photopsin splits into retinal and opsin. So we already know that both this rhodopsin and photopsin is composed of retinal and opsin. So in the presence of light, both rhodopsin and photopsin splits into retinal and opsin. So here also it splits into retinal and opsin. So as a result of this chemical change, impulses are generated and these impulses get transmitted through the optical nerve of our eye to the cerebrum, and this makes us the vision possible.